Welcome to my presentation, Translation in Prokaryotes. Translation is the first and most important part of protein synthesis. Ribosome catalyze the joining of amino acid monomers directed by the mRNA sequence. It is the mechanism in which codon of mRNA translated and specific amino acids in sequence form a polypeptide on ribosomes. The requirement for translation process requires amino acid, mRNA, tRNA, ribosome, GTP, magnesium ion, translocation enzyme, and release factor. Amino acids required for the synthesis of polypeptide chain. mRNA, which carries the message from DNA, and according to message, the protein gets synthesized. tRNA acts as an adapter and brings the new amino acid to join the polypeptide chain. Ribosomes are also called as protein factory and they join the amino acids into polypeptide chain. GTP is a source of energy which is required for the translation process. Magnesium ions, they assemble the smaller and larger unit of ribosome. Translocation enzyme required for the translocation of ribosome on mRNA and release factor which are required for the termination of process translation. The amino acids form raw material and these amino acids are 20 different types. These amino acids are also called as essential amino acids because these takes parts into the protein synthesis. All these amino acids are comes from the diet on the digestion of protein into amino acids and these digestible amino acids are present into the cytoplasm. mRNA serve as an intermediate molecule between DNA and protein. Ribosomes serve as site for protein synthesis. Each ribosome consists of large and small unit. The larger unit is made from 50S and smaller subunit is 30S. This subunit during resting position, these subunits occur separately in cytoplasm. This is the structure of ribosome. Here is a smaller unit of ribosome that is the 30S and 50S. This is the larger subunit of ribosome. The large subunit has three chambers. First is the A site where the amino acid tRNA bind. Second is the P site where the peptidyl transferase enzymes are present and they form the peptide bond between the two amino acids. Here is the third E site where the tRNA exits the ribosome. Only during protein synthesis ribosomal subunits get associated together due to magnesium ion. There is only first tRNA that is formyl methionine amino acid complex which directly enters P site of ribosome. Ribosome protects polypeptide chain from the action of cellular enzymes, also protect mRNA from the action of nuclease. The mechanism of translation takes place in three stages. First, initiation, second, elongation, third, termination. In initiation, the following steps takes place. First is the activation of amino acid because these amino acids are in the inactive form in the cytoplasm. So there is the activation of amino acid and formation of amino acid tRNA complex. Each amino acid is activated in presence of enzyme amino acid tRNA synthase. Then each amino acid is attached to the specific tRNA molecule at 3' CCA end to form amino acid tRNA complex. Once the tRNA complex is formed, then next step is a formation of initiation complex. It takes place in three stages. First, the three initiation factor IF1, initiation factor 2, initiation factor 3 and GTP binds to small ribosomal unit. Initiator amino acid tRNA and mRNA are attached with the initiation codon AUG by its anticodon UAC through hydrogen bond. Here is the initiation process diagram. This is the mRNA which carry the message. On mRNA, AUG is always start codon or first codon. Here is a smaller subunit, that is a 30S. IF1 factor and IF3 factor is already attached to the smaller unit. Then the IF2 initiation factor 2 with GTP and first tRNA, that is formyl methionine tRNA, is attached to the start codon AUG by hydrogen bonds. Then the larger, after formation of initiation complex, the large subunit join the complex and forms 70S complete ribosome. And the if met formyl methionine tRNA residing in ribosomal P site. This is the overall initiation process. First, initiate with 30S subunit, then joining of IF1, IF2, IF3, GTP, then tRNA, first formyl methionine tRNA, and they form the complex, that is the initiation complex, and then larger subunit binds to the smaller unit. This bind this bind to the smaller unit, then this tRNA comes into the P site. A site is empty, and here is all process is about initiation. Then after the initiation, there is a elongation of polypeptide chain. Elongation begin with the binding site of second amino acid tRNA at the ribosomal amino acid A site. The tRNA is escorted to the A site by the elongation factor EFTU, which also carries two bound JTPs. These JTPs is utilized, hydrolyzed, and energy gets utilized. After binding of tRNA, EFTU is released. A peptide bond is formed between the carboxyl group of terminal amino acid, that is the formyl methionine in the first cycle at the P site, and the amino group of newly arrived amino acid at the A site. This reaction is catalyzed by peptide transferase activity of the large ribosomal subunit. After EF-GTP binds to the ribosome, the GTP is hydrolyzed. The tRNA carrying the elongated polypeptide chain translocates from A site to P site. The discharged tRNA moves from P site to E site and leaves the ribosome. Consequently, the next mRNA codon is moved into A site, which is open for the next amino acid tRNA. These events are repeated amino acid, for each additional amino acid. Here is depicted by the diagram. This is mRNA from 5' to 3'. Then smaller subunit of ribosome, larger subunit. Here is a P site and A site. In the P site, there is a first formyl methionine tRNA. A site is empty. The second amino acid tRNA is enters into the A site. After the entrance, this formyl methionine amino acid gets joined with the alanine, that is the second amino acid, by peptide bond, and this activity takes place with the help of enzyme peptidyl transferase. Then ribosome shifts one codon in 3' direction. As a result, this alanine tRNA is enter into P site. This third amino acid enter into A site, while the first formyl methionine tRNA gets empty and exit from the ribosome. So this is the elongation process. Then termination and release of polypeptide, that is third and last step. At the end of mRNA, there is a stop codon UAA or UAG or UGA. It is exposed to the A site. It is not read and joined by any anticodon of any tRNA. Release factor binds to the stop codon, thereby terminating the translation. The polypeptide is now released in the cytoplasm. The two subunits of ribosome dissociates and last tRNA is set free in cytoplasm. Finally, mRNA is also released in the cytoplasm. Here is the diagram for the termination. Ribosome translocate up to the end. When there is a stop codons are present, either UAG or UAA or UGA, the release factor gets activated and it releases the free polypeptide chain. This empty tRNA. After dissociation, this mRNA gets digested or degraded into the cytoplasm, and both this unit, that is the smaller and larger subunits, gets dissociated. It requires energy in the form of 2 GTP. Thank you. For the student